This video will continue our discussion of molecular evolution. When considering evolution at the molecular level, a common question that is asked is whether variation in gene sequences are due to mutations favored by natural selection or just random genetic events. Mutations that are favored by natural selection are non-neutral, meaning they affect the phenotype of the organism and are acted upon by natural selection. According to Darwin, this is the dominant force in changing the composition of populations and leads to the variation in life that we observe. Of course, the complexities of our genomes was unknown to him, and thus he didn't have sort of all the information that he needed in his day. So that brings us to a newer idea proposed by Motu Kimura called the neutral theory of evolution. This theory states that most genetic variation in populations is due to the accumulation of neutral mutations rather than adaptive mutations. A neutral mutation is one that does not affect the phenotype of the organism and thus is not acted upon by natural selection. The spread of these neutral alleles in a population is governed by their frequency and by genetic drift. Since this theory is a bit of a contrast to Darwin's, it's sometimes referred to as non-Darwinian evolution, or more colloquially, survival of the luckiest. Now, to be clear, Kimura was not arguing that natural selection is not responsible for adaptive changes in species, simply that a lot of variation in gene sequences is due to neutral changes that eventually become adaptive once they have accumulated. To explain this neutral theory of evolution, Kimura and his colleagues suggested five principles that govern the evolution of genes at the molecular level. We will discuss those here. So let's go over each of these principles and an example of each. The first is that neutral substitutions in amino acid sequences happen at a roughly constant rate. We saw this earlier in the globin gene family, where there were amino acid substitutions, but they did not affect how the globin protein ultimately operated. The second principle is that proteins or parts of proteins that are less functionally important will accumulate amino acid substitutions more rapidly than important proteins or regions. We see multiple examples of this in things such as histones, which tolerate very few mutations. Additionally, amino acid substitutions in active sites of enzymes, that is, the part of the enzyme that interacts with the other molecules, are very rare. A third principle is that amino acid substitutions do not disrupt the existing structure or function of the protein are more common than those that are disruptive. We see this in several examples that are listed here, including mutations in the wobble base being more frequent and introns evolving faster than exons. A fourth principle is that gene duplication often precedes the emergence of a gene having a new function. Gene duplications lessen the selective pressure on genes, allowing one copy to mutate without consequence. Once enough neutral mutations have occurred in this gene, it may have a new function. The globin gene family is an excellent example of this, as each copy of the globin gene has a slightly different function in our bodies. Finally, the fifth principle is the selective elimination of deleterious mutations and random fixation of neutral mutations, or very slightly deleterious mutations, are much more common than the selection of advantageous mutations. This harkens back to the principle that most mutations that affect phenotypes are negative, whereas just a few of them are positive. Ultimately, these few positive mutations drive evolutionary change, but such occurrences are rarer than when compared to the opposite. Sequence data to date has supported Kimura's five principles, but The debate is, of course, not yet resolved, and scientists are still working out some of the details on these principles. One discovery that has cooled the debate is the concept of nearly neutral mutations. These mutations, uh, these nearly neutral mutations bridge the gap between neutrality and selection by showing that many mutations have small fitness effects whose fate depends on population size. This more unified explanation matched the empirical data far better than either pure neutrality or pure selection, effectively cooling the debate about the neutral theory.